lots of people ask me, and it's very confusing how New York Structural Biology Center is structured. It's a center of centers of centers. We have centers and centers. So Simon's Machine Learning Center is inside of the Simon's Electron Microscopy Center within New York Structural Biology Center. We like centers. And I'm the leader of the SMLC. So to give you a quick overview of what I'm planning to talk about today, I'm going to start with like an extremely brief overview of object detection, semantic segmentation, and instant segmentation, and sort of where these fit into the cryo-EM pipeline. I mean, you guys have already heard lots about cryo-EM and many of the different problems and like stages in cryo-EM data analysis. So I don't think we need to go really deep into that, just very briefly. And then I'm going to tell you guys about two projects in my group. So the first is looking at how we can basically use pose invariant neural networks to do unsupervised learning of objects, unsupervised learning on objects that have like different pose and locations and images. And so how we can actually make a network that is invariant to, learns content vectors basically that are invariant to pose, and is also simultaneously does inference on that pose in an equivariant way. And I'll show you some results on this in just general object representation learning, and then some specific application of this to cryo-EM for particle picking, which I think is hopefully an exciting new direction for thinking about how we can do particle picking in an unsupervised way. And then I'm going to talk about a geometric point cloud transformer for instance segmentation in micrographs and tomograms. And so here, fundamentally, the problem that we're looking at is that like many biological structures are not just represented as like a point in a micrograph, like a single particle, right? There are filaments and membranes and large organelles and all these kinds of things. And so oftentimes we end up getting some sort of, after doing some sort of semantic segmentation of a micrograph or a tomogram, you end up with like some large mass that you then turn into some sort of point cloud, and you have like a gajillion filaments in there, and you want to actually know, well, which of these are individual filaments and how can we solve that problem, which actually is quite difficult. So we frame this problem as this graph learning problem, and we train a neural network to actually do inference on this, where we're taking point clouds as inputs. And so I'll show you some empirical results there, and then I'll show you our end-to-end pipeline for microtubule annotation and tomograms that combines this with also a semantic segmentation network. So, okay, what are object detection, semantic segmentation, and instance segmentation? So in a cryo-EM context, I would break this down as object detection is like the problem of identifying like one or many specific objects in an image. So particle picking being like the example of this for cryo-EM. So in this problem, basically what we have is an input image, and we want to get out something like a bounding box or like a coordinate of the centers of all of these like individual, very discrete objects. Semantic segmentation is more like the problem of saying, okay, hey, I have this micrograph with all of these membranes in it. Can I just get a mask out, say like take this image as input and then have as output another sort of image where in that I would have for each pixel, whether it is background or of my semantic class of interest. And if you have multiple classes, you can make that multiple classes. But where fundamentally you don't know which instance of an object this belongs to, just like this is membrane or not membrane. And then instance segmentation is the problem of saying, okay, now I have all of these things maybe that I know are membranes, but which ones are the actual individual contiguous membranes in this micrograph, right? And so here in this case, there's a lot of them and they're extremely overlapping. And so going from say a semantic segmentation to an actual instance segmentation of this is a non-trivial problem. Because if you were just going to do something like watershed or some sort of like flood filling algorithm on this, you're going to say like, hey, this is one giant object here because it's contiguous in this sort of mask. But in fact, we know there's many, many, many membranes within this. Okay, so where does this actually fit in single particle cryo-EM? Grossly oversimplified. Basically we do some sample preparation, actually image, you end up with these large micrographs that have your particles embedded in it, and then you actually need to do some sort of analysis of these micrographs to say where are my particles or my filaments or my membranes, before you can actually go on and do what the exciting downstream stuff of like reconstructing your protein and figuring out all of its conformations and all of these things, right? So really, this problem of particle picking, this object detection problem is fundamental, is really fundamental because you need to actually identify all of these things in order for you to do anything with this data. So I'm going to start with talking about object detection here. And this is all work with my postdoc, Ali. So this is unsupervised pose invariant representation learning with the geometric variational autoencoder. And this all work is also appearing at NeurIPS. So if you're going to be there, you can meet my postdoc there as well and learn more about it. So as a really large big picture overview, basically we have a framework that allows us to do completely unsupervised learning, where we take a pile of images that contain objects where they have some unknown rotation and orientation. We have an encoder that's going to do inference on all of those properties plus what actually is in this image. We then have a decoder that's also equivariant to the pose and takes this content and then actually regenerates our image. And so we can use this to then do things like analyze our particles and make nice maps of the sort of content latent. It also is extremely accurate actually for doing inference on the orientation, even though it's completely unsupervised. And so then you can also do things like conformational analysis in 2D here. So in natural images, objects often have unknown orientations. This isn't only a problem in cryo-EM. In general, I think we tend to think about pose as not being some sort of intrinsic property of an object, right? It doesn't change its nature. If you have a protein structure, who cares how it's oriented in space? It's the same protein, it's in the same structure, and that's what matters. So really, we tend to view pose as this kind of nuisance variable that doesn't really matter for our understanding of the underlying object. However, in sort of any kind of naive analysis, pose oftentimes actually explains much more of the variance in images than content does. So if you take a stack of a bunch of particles in random orientations and you just try to train a variational autoencoder on it, or you do PCA on it or something, what you'll find is that oftentimes the main source of variation actually is the rotation of that thing and not sort of more subtle conformational differences or something like that, right? And so the question is, how can we learn about objects without knowing their location in pose? So basically, we want to actually be able to disentangle these multiple sources of variation in these images that we actually observe, right? So we have some content. That content is placed in some location, right? That's its translation, and it could be rotated as well. But fundamentally, we don't want translation and rotation to then somehow end up in our understanding of the content. So what we're going to look at here is a variational autoencoder framework for doing this. And you guys have now already heard lots about this and things like variational autoencoders and CryoDragon and other methods. So I'm not going to go into this in any great detail, but basically this is a very common technique. And really what this fundamentally boils down to is we're going to take some input image. We're going to have an encoder network that does amortized inference on some sort of latent vectors that we are using to describe what this image is. And then we have a decoder that takes those latent vectors and tries to actually reconstruct the image. And we train this thing end to end by feeding in an image, doing inference on some latent variables, feeding that through the decoder, getting our output, and then minimizing the error between the output and the input. And so what's really important in these frameworks is how you actually choose the encoder and the decoder, right? This is the secret sauce here is what neural net architecture or algorithm am I going to use to do this? So I'm going to start by talking about a decoder. And so hopefully this won't be boring since you guys have already heard lots about the CryoDragon decoder. But basically, we can choose a decoder that is equivariant to any coordinate transformation by parameterizing an image sort of through this neural network, right? So we have a neural network. You feed in a coordinate of an image plus, in addition, some latent variables that represent what actually is in this image. And if you have a neural network like this, you can do all kinds of fun transformations on your coordinates, and your output image will transform in exactly the same way, right? So because we're decoding an image from actually evaluating this function on the pixel coordinates, so if we rotate the coordinates, we get a rotation of the image. If we translate the coordinates, we get a translation of the image. And it's completely equivariant, right? There's no sort of need to do any interpolation or anything like that either, right? if I translate my coordinates by delta x, my image will translate by exactly delta x, and I don't have to somehow like train my network to do that. It just does it, right? Okay, so great. So, well, how can we think about encoders? Okay, so, well, encoders usually, and so like what I'll talk about here is like, this is basically how an encoder is structured in most of the methods these days, including CryoDragon, and this like goes back to some work that we looked at looking in 2D at basically using this kind of decoder to learn properties of 2D images, right? But what we're doing here is we're using an encoder that doesn't have the good properties that our decoder has, right? So like our decoder here has these wonderful equivariance properties to coordinate transformations, but our encoder is just some fully connected, sorry, I was pointing at the decoder, but I should have been pointing at the encoder. But our encoder is actually just some like fully connected neural network here that takes this image as input and outputs the parameters of our approximate posteriors. And so if I do some translation of my coordinates, like if I translate this image, well, yes, this network is equivariant to like the translation of the input coordinates, but this network is not. So there's no guarantee that this network will tell me if I translate this image by delta x that actually, oh yeah, this output should also change by exactly delta x. Absolutely no guarantee of that. So as we know, these VAEs actually work great for inferring the semantic content, right? So as you know from CryoDragon and other methods, right? They work really well for inferring this latent variable. But there's a reason why, until recently, right? Like there haven't been good methods for inferring these latent variables, and it's because these networks do not work well for this at all. So to show you a quick example, when we were first looking at this work, right, with spatial-VAE, basically, if you take even like a really simple data set like MNIST, and you rotate the digits uniformly. So I'm just going to take all of the digits in this data set, apply some random uniform rotation to them, apply a little bit of translation, and try to train this framework in sort of the naive way. It completely fails to learn the rotations. Completely fails. Just because this network is not good at this problem, right? It gets trapped in some very bad local optima and just cannot learn this. Right, so this predicting rotation is inherently a non-convex problem. That's one part of this issue. And the other is just that, and the other is that our network isn't equivariant, right? So if I rotate my image, my output doesn't rotate, and therefore it doesn't have nice learning properties, it doesn't have good inductive bias. So, okay, so we've been thinking about sort of a different approach to how we can solve this problem, right? So one thing we can think about that's actually really nice here is translation's not a very hard problem because we have convolutional neural networks that are translation equivariant. Right? So naively, a convolution is translation equivariant. If you have some image and you apply some filter to it and you get some output map from that filter, well, if you translate the image, then your output map is going to translate exactly the same amount. But if you rotate the image, your output map is not going to rotate the same amount because the component is not rotation equivariant. So what we really need is a convolutional neural network that we can use as our encoder that is both invariant, that is equivariant to translation, but also to rotation. So that's the challenge. So enter group convolutions. So there's been quite a bit of work in sort of the ML field on thinking about how can we actually design convolutional neural networks that are invariant to rotation and some other interesting transformations like flipping. And actually, ultimately, what this boils down to is a really simple idea, right? So a convolutional neural network is equivariant to translation because you basically apply this filter to every window of your image. So why don't we just do the same thing with rotation, right? Okay, I can take my image, I can form k rotations of that image, I can feed all of them through my network, get some output, and then I can basically just extend my convolution to also now convolve over that like rotational space, right? So basically, we can just enumerate some discrete set of rotations, so in this case, say like 4 rotations, apply our network to those, and then also now apply our network, we can actually now convolve over this sort of like rotational space where we basically converted the rotation of this image into some sort of like the phase in this feature space, right? So if I rotate my image, then I actually end up with like a phase shift of 1 here. Of course, this is discrete, but so are pixels for translation, so maybe we can make a sufficiently fine-grained rotational space, and it'll be fine. And then we can actually design a neural network that's completely equivariant to rotation, because now we've converted a rotation into this sort of like shift in this like rotational feature space. And so we can now build a neural network encoder by taking advantage of this that is now sort of both rotationally and translationally equivariant. So the idea is basically if we have an encoder now that I'm going to scan over basically all translations and rotations of every sort of patch of my image, and then take those outputs and somehow use those outputs to then do inference on translation and rotation, then all of a sudden, if I translate my object, my output will translate that much. And if I rotate my object, my output will rotate that much. And it's guaranteed. I don't have to train my network to do that.
I have a question.
Yes.
If you want to use this for 3D reconstruction, wouldn't it be more like because you're trained? So just simple like 3D reconstruction. I actually, if my network outputs a translation each time my center is misplaced or something, a little bit more complicated. Like, doesn't it complicate the 3D reconstruction problem? Because you're
so for 3D, you need to do, yes, so here I'm talking about only in-plane rotations, right? For 3D, you would have to consider how you could do this for SO3, which we're working on and actually have a solution to, but it's, not available yet. But yeah, we can do it. We can do it too. But yes, it is more complicated because of that. I mean, and in principle too, it makes the complexity worse, right? Because you have a higher dimensional space that you now need to sort of like tile with things. But what we've seen in 2D that maybe saves this is it turns out that like as long as you're sort of like close enough, all of a sudden it can work. And I think it's because, well, maybe we can talk about that a little bit more later in the talk. But yeah, good question. Okay, so now what we're going to do with this network is we're going to break down sort of our inference process into doing inference on like these three properties, right? So given an image, I want to do inference on its location, its rotation, and this content vector. And something important to consider here is my network is outputting sort of like a potential for each location in my image from my convolution that my object is there. And my network is also outputting a potential for each of these discrete rotations that my object is there. But especially for rotation, we would have to define this rotation to be very fine-grained for this to be accurate. So what we're going to do instead is we're going to, actually answering exactly that question now, is we're going to rely on the idea that as long as we can get close enough, rotation becomes convex, right? So estimating rotation as non-convex, like if you have an object and you rotate it 180 degrees and then you calculate the loss of that, well, your gradient's not going to be very informative in terms of getting you towards the global minimum. But if you only rotate, say, like 5 or 10 degrees, maybe you're close enough that now all of a sudden your gradient is actually pointing in the right direction. And so from that perspective, as long as we have a sort of coarse enough representation of our sort of discrete set of rotations, then now maybe our network can actually accurately do inference on like some sort of like delta of that to tell us exactly what the rotation of our object is. And we can train that by gradient descent because it's close enough that that will actually be accurate, right? It will actually fall into sort of the local optima and predict the correct rotation. And so what we have then is we basically have formulated this as a mixture model where given our discrete translation and rotation outputs, we're going to then conditioning on those, we're going to also predict sort of a fine-grained edit to the rotation. And we're also then going to predict our content vector. And because our content vector is conditioned on the location and rotation of the object, our content vector becomes invariant to rotation and translation. So putting this all together, we basically have this framework now of this rotation and translation invariant or equivariant encoder that's doing inference on all of these parameters, and our decoder is also invariant to these things. And so now our whole framework is very nicely invariant to rotation and translation, and we can just train it. So what I'm going to show you first is a proof of concept of this on MNIST. So I motivated to you that MNIST is surprisingly hard when you make uniform rotations of it because like a naive encoder will completely fail. In fact, the only way you can get a naive encoder to work is if you don't rotate your digits uniformly, right? Like if you have some sort of bias here, then actually it can kind of figure it out because like, oh, it's mostly in this sort of sync and it does okay. So we're going to start by looking at these two problems. So the data set that's either uniformly rotated or rotated with a bias. And then, and so the whole training framework here is completely unsupervised. So I'm going to train these models. And then what I'm going to show you is if we actually take our inference network and we predict the rotation, how accurate are we? And then to understand like how good our content vector here is, we're just going to cluster our images. We're going to take our corrupted images, cluster them by that content vector, and then just say like, OK, do we correctly predict what the digit is, basically? Like, do those clusters match the actual underlying digits? So to start, I'll show you the baseline here, which is this VAE with the naive encoder. And you can see it actually kind of figures something out when your digits aren't randomly, uniformly rotated. It actually has some correlation. It's non-trivial here between the predictions and the actual rotations, but it fails, as I mentioned before, on the uniformly rotated data set. But when we actually use this equivariant encoder, our results improve dramatically. So in fact, on the normally rotated data set, all of a sudden we have a huge improvement in our ability to predict the rotations. But also it actually starts to work now on the uniformly rotated data set and also actually predicts the rotations very accurately. And just to show you, this encoder is rotationally equivariant. So as we rotate this digit and feed it through our encoder, our actual predicted rotation moves exactly as we expect as we rotate the digit. So there is sort of no guesswork or like wishing that your network is invariant here, right? It's not going to do something funny. It is exactly equivariant. Oops. So you might be asking, hey, what's going on here? That doesn't look so great. Well, it turns out there's actually is ambiguity in digits in their rotations, right? If I show you a 1, well, it could be rotated by 0 or by pi. Right? There's clear symmetry there. And so actually, this is due to this kind of symmetric ambiguity. So when we assign some arbitrary rotation to this, and the network is telling us actually it's rotated by pi from that, but really they're equivalent. And so that makes, that's what's going on here. So in fact, the network is performing quite well. And you can understand this better if we break down these plots down by individual digits. So like 1, has these very clear striping effects. 8 does too, because it's also roughly symmetric. And there's, of course, some error here and there otherwise. But largely, it does a very good job.
So in these pictures, so you discretize the circle somehow, right? I mean, you picked a certain number of angles, like when you did the convolution.
Yes.
Yeah. So how many, like in this pictures, how many?
This network is using 8.
8 sample.
But I'll show you a comparison of that in a second.
And also, could you actually not use the laser pointer? It's kind of.
Oh, is it?
Yeah, it's a little distracting.
I'm sorry. Okay, sure. I'm waving around. Is it like jerking around too much? Yeah.
So one can show up like in different angles. So how do you determine like what's the reference?
Good point. Yeah. So the reference angle is because we rotated the digit ourselves, right? We applied some random rotation to it, and so the reference angle is what our network predicts for the original input. So we're actually measuring the difference in those angles. Because we don't know that reference. Exactly, yeah. Sure.
So do you also encode what digit it is in the latent space, and the decoder fully generates, has a generative model for what like a two-took looks
Correct.
like?
Yes, that's how we're going to cluster them, which I'll show you in a second. Yeah. Okay, so let's look at clustering, right? So then the question is, does this actually improve our ability then to actually understand what these digits are in this like semantic latent variable? And the answer is yes. And also what I can show you here is that our representations are now invariant to the rotation and translation. So if we change the digit, we move in the content space. But when we rotate that digit or translate that digit, it doesn't change now what our network thinks about the actual like semantics of this digit. Right? Yeah.
So if we put this through the spatial-VAE,
Mm-hmm.
the original one, what would happen? So if we had some latent space representation and we started rotating, what would you see?
It would move.
It would move around.
Yes. So I mean, Roy was talking about this the other day, right? So the Cryo-Dragon encoder is just sort of this naive neural network, right? And that's why when you rotate your particles, it moves in the content space. Same problem here. Yeah.
Where are 6 and 9 here?
They're
You can see on the
here
Oh, okay.
and here. So yeah, you can see there's some overlap because yes, there's a bit of ambiguity between them when you rotate them. Yeah. Oop, okay. So right. So if we actually then break this down and look at the effects of some of these hyperparameters that we were just discussing. So yes, one question was, okay, how much does it matter how finely we sort of tile rotation space? And so if you look here, we're looking at 3 networks with different levels of how fine-grained we tile the space. So we're looking at 4, 8, and 16, sort of breaking rotations up into 4, 8, or 16 discrete steps. And as you can see, actually, if we go from 4 to 8, we do get a pretty big improvement in our ability to, in this case, we're looking at clustering accuracy, to then cluster the digits. And there is actually some improvement still in going to 16, but it's not that big. And so with this, of course, we do expect there to be some diminishing returns here because our network does have this other component that allows it to also sort of now guess at, okay, if I'm close enough, it can sort of find that delta offset to get the correct rotation. Right. And so if we compare this to what this sort of clustering would look like if we ran it through the vanilla spatial-VAE, that's what it would look like. I don't have an animation to show you how it would move if we changed as we rotate digits, but you get an idea here that very clearly the network is much worse at actually distinguishing the objects.
So this is a stupid idea, but then why does Cryo-Dragon work? Like in the sense, like if you have this mixed up representation of content, like why do you in the end you can see things?
Well,
like
there's a reason that the original version of Cryo-Dragon does not do inference on the rotation and translation. Yeah. It's not because we didn't try it. It's because this is a hard problem to solve, and it doesn't naively, like it doesn't immediately work, right? You have to try all kinds of tricks and like things to try to get this to work, right? And it's a hard problem. But yes, it is interesting to me too that the semantic inference does work. But maybe it's because those things are close enough, and so it's okay if it ends up getting encoded into like a slightly different conformation of the protein, but it's close enough and you end up, and it averages out all in the end or something. It's not sure. But it probably does impact the like convergence speed of the network, right? You could imagine that if you were doing that inference better, you probably would converge much faster in training the decoder. Okay, so I'll just show you quickly a quick ablation study just to try to prove to you that actually this multimodal sort of rotation structure and also having actually using a convolution to do inference on the translation is necessary. So if we were to just not use our like rotation group network here, and we were to only sort of just try to take advantage of the translational equivariance of a convolutional neural network, this would not work. This is what the kinds of results that we would get. And also, even if we use a group convolution here with rotation inference, but then we sort of like pool over the rotations or then apply some sort of fully connected network on those representations, this also doesn't work. It's really critical to actually fully take advantage of the equivariance that we get sort of from framing those outputs as these like potentials of your object being there. Okay, so what can we do with this in cryo-EM? So one thing I think is really exciting to think about here is like, okay, this is a way that you can do things like fast 2D classification, aside from applications to 3D, of course, because in 2D classification, you still have the problem of in-plane rotation, and you can understand things like the conformations and views of your particle, and cluster those. And we can then, of course, look at things like continuous motions in 2D. But this clustering implies something kind of interesting, which is you might think, well, okay, what happens if you actually have patches of images, like patches that don't contain an object, right? So we've made a very strong assumption in this model, which is that every image contains exactly one object. And so you might ask, maybe this is too strong, sure you can train it on a particle stack, but actually it works surprisingly well if you just train it on like random patches of a micrograph. So what we've been doing now is just training this thing on random just micrographs where we just chunk it up into random patches and train it and see what does it learn. And it turns out actually it's surprisingly good at localizing particles just from that naive application, right? There's no reason necessarily to expect every patch will contain a particle, but enough do. It's close enough that it actually works pretty well. So I'm showing you here is just a micrograph on the left, and hopefully you can sort of see this activation map on the right, which is like the probability that there is a particle at this place. And it matches surprisingly well, and it actually is quite good at actually identifying where the particles are. And this is completely unsupervised. So with that in mind, sort of the conclusions and next steps here is basically, what I hope I've convinced you of is that by designing a model here that actually captures these relevant equivariances, we're actually able to much better disentangle sort of our content from these nuisance variables, and also then are able to actually do accurate inference on those nuisance variables. This allows us to then, of course, do things like amortize and accurate pose inference. And so in the future, I think there's some really interesting ideas here around, okay, right, maybe this can underpin some next generation of particle picking algorithms, right, where we can actually potentially do some sort of fully unsupervised particle picking. And then, 3D equivariants, right? So how do we actually start to take networks like this and use them to actually do full, make them like SO3 equivariant, to use them for inference of pose and reconstruction. So yeah, those I think are some exciting next steps. So now I'll tell you a little bit about sort of part 2 here, which is semantic and more actually instance segmentation and applications, how we can sort of try to build networks to be able to do instance segmentation of like complicated situations like this, so that we can actually do better membrane and filament and hopefully also things like organelle analysis in the future. So this is a geometric point cloud transformer for instance segmentation in micrographs and tomograms. So so I already told you about sort of what semantic segmentation and instance segmentation are, so I won't belabor the point here, but so the problem here is we have a micrograph, we want to do some sort of annotation of where membranes are, but then what we really want to know are what are the individual contiguous membranes in this micrograph. And if we're looking at something like a tomogram where we have microtubules, where we have a tomogram, we can do some sort of semantic segmentation of where are all the microtubules here, like what voxel is in a microtubule, but then what we really care about are, can we actually then identify and like extract each of those individual like contiguous microtubules in this data so that we can do some sort of downstream biophysical analysis or something like that. So what makes this hard? You might just say, but hey, this is just like curve tracing or something. That's not that hard. But these objects are complex, right? They can intersect and overlap. So it's not obvious from a segmentation mask, what are the individual objects here. There can also be errors in semantic segmentation, right? Semantic segmentations give false positives and false negatives, so there could be gaps. Existing methods have a really hard time tracing objects in these kinds of crowded environments. So like there's tools and things like Amira for doing this. There's like multi-curve fitting, but they have a very hard time in crowded environments like this. So what we're going to do is think about this as a graph learning problem. So basically, if we have some point cloud that's like, here's locations in membranes or locations in filaments, right? Like that tiles all of my data, but I don't know which points belong to which individual object instances, then we can think about this sort of instance segmentation problem as a graph prediction problem. So I want to take this point cloud, and what I want is a graph where for each pair of points in my point cloud, they only exist in the same sort of like connected component of the graph if they're in the same object, right? Like every object should be a connected component of the graph. Because one of the other challenges here is we don't know the total number of objects. So we need some method that can identify an arbitrary number of objects, and so this would be like one way to formulate that. So what we're going to do is formulate learn a function, so learn a neural network that's going to take this point cloud as input, and it's going to output for each pair of points the probability that an edge exists between them. And then we're just going to do a graph cut, and we'll have our instances, right? So we're calling this the dimensionless instance segmentation transformer, because the idea is, given this input point cloud, we're going to first convert this into just a distance-based representation. I'm just going to calculate the distances between every pair of points, and then derive some like feature vector from that, and that's going to be the input to my network. And what that means is that if I sort of rotate this point cloud around or translate this point cloud around, it doesn't change the representation that goes into my network, so my network will be completely invariant to those things, which we want, because that shouldn't matter. And it also means we can apply this to sort of arbitrarily high-dimensional point clouds, right? This could be 2D, fine, we can calculate distances. It can be 3D, because we just calculate distances. And so in that sense, like you can apply this to 2D, 3D, 4D, 5D, whatever data. So then we're going to take this distance matrix, and we're going to process it through a neural network that is sort of very similar to AlphaFold, where you have this pair representation. So what we're going to do is we're going to do these updates, attention-based updates on this pair representation, where we try to have a network that's inspired by sort of like the triangle inequality to preserve something about like the fact that distances should preserve that, and so like the representations between points should update in that way. And then eventually what we'll get as an output is now for each pair of points this probability that there's an edge between them in our like instance graph. And we're going to train this on labeled instances from membranes and microtubules that we actually have like labeled the instances on. Okay, so it turns out this dramatically outperforms actually other approaches for doing this. The network works amazingly well at this problem. So what I'm showing you here is, this one's a little hard to interpret, but basically in red is the ground truth, and then in green is our predictions, and so yellow here is overlap. And to sort of quantify this, we can look at like the mean average precision of these assignments, and this dramatically outperforms like multi-curve fitting or just like a distance-based approach for trying to assign the individual instances. And if we look at this in, let's see if I can play this video, in 3D on microtubules, maybe it will play at some point. Then, here you can see like Amira is the sort of typical tool people would use for this to label their microtubules. And so here's sort of what like a raw point cloud would look like here. So identifying the individual microtubules is really hard. But, doesn't like the video. Okay, but we are able to actually much more accurately identify the individual microtubule filaments here in this data. And so with this point cloud transformer. And so what we can then do is, since we've gone and put this into sort of a whole end-to-end framework for actually doing microtubule analysis, so the thing is, this doesn't solve the semantic segmentation problem, right? We're only doing instance segmentation. So we have another network for doing the actual semantic segmentation part. And then if we put this all together, basically we can then do end-to-end, fully automated semantic and instance segmentation of microtubules. And I'm showing you an example here of this applied to a heliosyl, so in situ. So step one is to do this kind of semantic segmentation. And you can see here, as this tilts a little bit, that the semantic segmentation network is quite good, but it definitely has some like false positives in some places around the edges, and there will be breaks in this network. And so we then convert this to a point cloud and run the instance segmentation model. which then sort of removes a lot of those false positives and actually is able to read over some of those gaps and things like that, so that we end up with like a nice instance segmentation now of all of the microtubules in this tomogram. And just so you can actually see too, like how many individual microtubules there actually are in here, you can color it by those instances, and you can see. So there's a lot of them. So we've and we can separate all of these. Okay. So quick aside is, yeah, you can apply this to other instance segmentation problems for like LiDAR data and other point clouds, and it works pretty well. Okay, so to conclude on this part, instance segmentation of membranes, filaments, and other large objects in micrographs and tomograms is hard, but it's really important if we actually want to be able to understand sort of the biophysical characteristics of these objects, right? If you want to understand things like the curvature or the length or any sort of systematic analysis of those kinds of properties, you need to actually be able to extract all of these individual instances. So by structuring this as a graph learning problem with our translation and invariant and rotation invariant network here, we're able to dramatically outperform the state of the art in 2D and 3D instance segmentation. And so we've now built this into like a complete framework for microtubule analysis, but we're planning to extend this to other things like membranes and organelles and other filaments. And actually, the instance segmentation network generalizes surprisingly well. So one thing we were really surprised to see was that if we trained it on the 2D membrane data, it actually performed shockingly well on the microtubules, even though it was not even trained on 3D data. So the actual instance segmentation network is pretty general, and you can just sort of plug and play it with some new like semantic segmentation network for labeling other filaments like actin or whatever your favorite filament is. And it's available for beta testing. We haven't made it public yet because we're still working out some issues with it. But if anyone's interested in trying it, let me know. Okay, so since I have you here, I just thought I would drop one slide on you about some other work going on in my group. So one thing we've been working on a lot recently is sort of automating actually the data collection process for cryo-EM. And so we have a tool called Ptolemy, which is now also integrated with Leginon and Smart Leginon to drive automated data collection, which you can use to actually identify like squares and holes and do, and also do fully automated screening. And there's some more work here coming. So with that, I'd like to thank the New York Structural Biology Center, everyone in my group, of course, Bridget and Clint, Alex Noble, Ellen and my PhD advisor, Bonnie, for all of their contributions to this work. And I think that we can do questions. And I'll